For our project, we chose to use autosomal recessive disorders to demonstrate the connection between Mendelian and molecular genetics. As you will see, the inheritance patterns of our two example diseases, Tay Sachs and Gaucher disease, abide by Mendel's laws of inheritance, and the biochemical mechanisms behind these diseases reflect the core concepts of molecular genetics. Although Tay Sachs disease and Gaucher disease have different underlying genetic causes, their phenotypes closely resemble each other. This similarity displays the diversity of the human genome and the profound effects of genetic alterations on the physical expression of genes.
Gregor Mendel developed 3 principles of inheritance from experimenting with pea plant breeding that describe how genetic traits are passed on. Mendel's experiments resulted in these main principles that rooted from the theory of heredity, where inheritance involved the passing of genes from parents to offspring. From this theory, he was able to create his principle of segregation, where during reproduction, each parent's pairing of genes split, and one gene from each parent goes to the offspring randomly. Another key principle that was created was his principle of independent assortment, where different pairs of alleles are passed on to the offspring independently and randomly of each other. There are exceptions to these principles, like how independent assortment does not apply to genes that are linked on a chromosome, and alleles don't always have a dominant/recessive relationship. There are also patterns of inheritance, like autosomal dominant and recessive patterns, X-linked dominant and recessive patterns, and as well as a mitochondrial pattern. Each of these affect people in different ways and are also present when looking at diseases. The next point is the chromosomal theory of inheritance and how it demonstrates Mendelian genetic principles. After studying the genetics of fruit flies, the chromosome theory of inheritance was confirmed from the data that was collected from Thomas Hunt Morgan. When studying fruit flies, he found a mutation that made flies' eyes white instead of their normal red color. Looking further into it, he found that the eye color was from inheritance and had different patterns depending on if it was a male or female. This type of inheritance pattern is a sex-limited one, and this gene is associated with the X chromosome, which is determined when crossing the flies to each other. The chromosomal theory of inheritance is a theory that genes are located specifically on particular chromosomes, and the behavior of these chromosomes during meiosis explain why genes are inherited. These chromosomes come in homologous pairs, one from the mother and the other from the father. Homologous pair members separate in meiosis, and the egg or sperm will have only one member. This process mirrors Mendel's law of segregation. Members of different chromosome pairs are separated into gametes that assort independently, and this process mirrors Mendel's law of independent assortment.
Now that we've finished up with that, I'm going to be talking about autosomal recessive disorders as an introduction for our two disorders that we will be talking about later. Autosomal recessive disorders are caused by inheriting two mutated alleles, one from each parent, and while they both come from each parent, neither of the parents' health are affected except in rare cases, and both have an equal chance of being a carrier. Recessive disorders tend to exhibit a horizontal transmission pattern, which means that inheritance can be random for multiple individuals in one generation to no individuals in the next at all. And recessive disease alleles typically code for less efficient proteins, if any at all, which a few examples of some of those disorders are Gaucher's, Tay Sachs, and cystic fibrosis, which we will be going over the first two in the next few slides.
Next, we are going to take a look at two autosomal recessive disorders, Tay Sachs disease and Gaucher disease. These two conditions reflect the genetic principles and patterns of inheritance previously discussed. You may be asking, how are Tay Sachs and Gaucher disease alike? Both Tay Sachs and Gaucher disease result from mutations of genes that code for lysosomal storage enzymes. These mutations cause a defect in the degradation of glycolipids, leading to a toxic buildup of these molecules in the central nervous system. The accumulation of these abnormal substances is the underlying cause of the neurodegeneration that characterizes the more severe forms of these conditions. Complete lack of the enzymes that break down the glycolipids produces a phenotype in which symptoms appear within the first 4 months of life, and severe neurological problems and death occur by age 4. Less severe or chronic forms of each of these diseases are caused by low levels of enzyme activity. Additionally, both Tay Sachs and Gaucher disease have been found to be more prevalent in the Ashkenazi Jewish population. The genetic cause of Tay Sachs disease is mutations in both alleles of the hex A gene found on chromosome 15. This gene codes for the alpha subunit of the beta-hexosaminidase A enzyme. This lysosomal enzyme is responsible for breaking down GM2 gangliosides, which are lipids found in the central nervous system. 78 mutations of the hex A gene have been linked to Tay Sachs, including 65 single base substitutions, 1 large and 10 small deletions, and 2 small insertions. Most of these mutations produce a completely non-functional form of the beta-hexosaminidase A enzyme. This visual shows the location of the hex A gene relative to other genes that cause different diseases. The hex A gene is located on the long arm of chromosome 15 at positions 23 to 24. There are three forms of Tay Sachs disease. The first is classic infantile, which results from almost a complete lack of the enzyme and progresses very rapidly. It is also the most common and the most severe. Symptoms develop around 6 months old, and patients usually do not survive past 3 to 5 years of age. The second is the juvenile form. Now, this form is very rare, and it is caused by very little enzyme activity, usually less than 1%. Symptoms develop between ages 2 to 5, and life-threatening complications occur around age 15. The last form is late onset Tay Sachs, which is very rare and is also the least severe. It is caused by less than 10% of normal enzyme activity. Symptoms can appear anywhere from late childhood to early adulthood, and lifespan varies from shortened to unaffected. Many of the symptoms associated with Tay Sachs are those of the infantile form, which is the most severe. But some of the symptoms listed above are observed in the juvenile and late onset forms as well. For example, individuals with juvenile Tay Sachs may begin to lose their sight and experience seizures. The symptoms of each form will be discussed next. The various forms range in severity, and the symptoms are not the same in each case. For the infantile form, the symptoms include psychomotor regression, seizures, development of muscle weakness, and eventually paralysis, loss of mental functions, becoming increasingly unaware of their environment, blindness, difficulty swallowing, and eventually death. The hallmark symptom of Tay Sachs is the presence of a cherry red spot on the eye. For the juvenile form, symptoms include frequent infections, behavioral problems, and these individuals have a more slowly progressive loss of movement control, speech, and mental function. They may also begin to develop seizures and loss of vision. For the late onset form, the symptoms include slowly progressing neurological impairment, which may lead to clumsiness and loss of coordination, muscle weakness, difficulty speaking and swallowing, and uncontrolled muscle spasms or movements. There is currently no cure for Tay Sachs disease. Symptoms can be managed with medications, physical therapy, respiratory care, and feeding tubes, but there is no treatment to stop or slow the progression of the disease. Currently, there are clinical trials investigating gene therapy and enzyme replacement therapy as potential treatments in the future. Additionally, there have been reports of substrate reduction therapy using the drug miglustat in bone marrow or hematopoietic stem cell transplantation. Substrate reduction therapy uses small molecules to slow the rate of glycolipid synthesis, which in this case would lower the rate at which GM2 gangliosides accumulate in the central nervous system. Enzyme replacement therapy has shown to decrease somatic symptoms, but since the enzymes are unable to pass through the blood-brain barrier, this treatment is less effective at preventing neurodegeneration. The gene therapies for Tay Sachs that are being developed use adenoviruses or adeno-associated viruses as vectors to deliver cDNA that codes for the alpha and beta subunits of the hex A gene. This approach is being tested in mice and Jacob sheep. Next, we are going to look at Gaucher disease, which is another autosomal recessive disorder. You may notice that Gaucher's disease type 2 has a phenotype that closely resembles that of the infantile form of Tay Sachs. The genetic cause of Gaucher disease is mutations on both alleles of the GBA gene, which is found on chromosome 1. This gene codes for the beta-glucocerebrosidase enzyme, which is a lysosomal acid. This enzyme normally cleaves the beta-glucosidic linkages of glucocerebrosidase lipids. Almost 300 unique mutations of the GBA gene have been described, including 203 missense mutations, 18 nonsense mutations, 36 small insertions or deletions, 14 splice junction mutations, and 13 complex alleles carrying 2 or more mutations in cis. These mutations result in a deficiency of glucocerebrosidase enzyme activity, leading to a toxic accumulation primarily in the liver, spleen, bone marrow, and central nervous system. This visual shows the location of the GBA gene on the long arm of chromosome 1 at position 21 or 22. Here you can also see the position of this gene relative to other genes that have been linked to cause other disorders. There are multiple forms of Gaucher disease. The first is type 1, which is also known as the non-neuronopathic. This form is the most common, and it doesn't involve the central nervous system, which means it is the least severe. It may appear early in life or even in adulthood. The second type is type 2, which is also known as the acute neuronopathic. This type occurs in newborns and infants, and it does involve the central nervous system. This form causes life-threatening problems beginning in infancy, and this is the type that has a phenotype similar to Tay Sachs disease. Type 3 is also known as the chronic neuronopathic, and it occurs during the first decade of life. It also involves the central nervous system, so here you will also see neurodegenerative symptoms. Patients may live into their teens or early 20s, but others have lived much longer into their 30s and 40s. Type 3 progresses slower than type 2. In addition to these 3 forms, 2 others exist: the perinatal lethal form and the cardiovascular type. The perinatal lethal form is the most severe and occurs in less than 5% of patients. Individuals with this form typically die before 3 months old or even in utero. The cardiovascular type of Gaucher's is characterized by the calcification of the heart valves, and it also involves the central nervous system. Complications often result in reduced lifespan, but some patients survive into early adulthood. We're going to focus on the We're going to focus on the first three types of Gaucher disease because the perinatal lethal form and the cardiovascular type are very, very rare. So the symptoms of the first three are displayed above. As previously mentioned, the lipids accumulate mainly in the liver, spleen, bone marrow, and central nervous system. So as you can see, many of the symptoms of each of these three types affect those parts of the body. Many patients with type 1 live their lives with minor symptoms or completely unaffected. Both types 2 and 3 result in reduced lifespan, and type 2 is the most severe, with death occurring between the ages of 1 and 3. In contrast to Tay Sachs disease, there are multiple treatment options available for Gaucher's disease. Symptoms can be managed using pain management therapies, blood transfusions, orthopedic surgery, or in rare cases, a splenectomy. There are now FDA-approved drug therapies, including enzyme replacement therapy and substrate reduction therapy. Enzyme replacement therapy has been shown to be an effective treatment for type 1 and the systemic aspects of types 2 and 3. However, this type of therapy does not reduce or reverse the neurodegeneration seen in types 2 and 3. Currently, there are a few studies looking into gene therapy for Gaucher's disease. One of these is working on a way to use retroviruses as vectors to deliver and insert functional forms of the GBA gene into abnormal or diseased cells. Trials in mice have had positive results. The inheritance and behavior of autosomal recessive disorders reflects the principles of both Mendelian and molecular genetics. Although each disease is caused by abnormalities of different genes, Tay Sachs and Gaucher disease have highly similar manifestations, treatment options, and clinical trials going on. The biochemical processes behind these diseases reflect the principles and concepts of molecular genetics. Tay Sachs and Gaucher disease are examples of genetic problems that may be solved in the near future through scientific investigation using the principles of both Mendelian and molecular genetics. That's the end of our video. Thanks for watching.